Welcome everyone. So today we have with us Associate Professor Silas Villas-Boas from the School of Biological Sciences. So Silas got his PhD at the Technical University of Denmark. He worked at Ag Research for 3 years and joined Auckland in 2007. So his research interests are primarily in microbial metabolism, metabolomics, and natural products derived from microbes. So since 2001, he's published more than 80 papers and 9 book chapters and 3 patents in this area. He contributed and co-edited the first international textbook on metabolomics, and he's been awarded the Early Career Research Excellence Award from the university for discovering certain novel bioactive metabolites. So I'm pleased to present Silas and talk to us about metabolic overflow.
All right, thank you. Thanks, Vino. Which screen should I focus on? They are quite apart from each other. I'll stay here because of the recording. So thanks for coming, and sorry for my late abstract. I missed the date from Vino. Before I start, I would like just to introduce my lab and what we do in SBS. I know most people here work in silico, in modeling, and I'm sure there are things that we do could help more on the wet side of systems biology. And I think metabolomics is a key component, mainly on thinking about metabolic modeling. My lab, I did my PhD focus on metabolomics when metabolomics was actually being conceived as an idea of looking at the whole set of compounds you can detect, not really identify, but detect in a biological system. Classically, analytical biochemistry focuses on a list of compounds that you look for, and disregard anything else. Metabolomics shift towards consider everything you see in a sample and separating what is artifact from what could be real signal molecules, a signal of a molecule, and consider that all to generate hypotheses. So metabolomics will never prove anything. We will create hypotheses, and through target studies you can validate those hypotheses if it's true or not. So it's a system biology approach. Instead of you have a problem, instead of generating hypotheses, you do an experiment, a comparative experiment, and use the data to generate hypotheses for you. And then you choose the hypothesis you think is more likely to, more relevant at least for your study, and then you can go and try to prove if it's correct or not. So it's just a different way of approaching science. My lab, what we have central to my lab is metabolomics tools. We spend a lot of time developing methods, most on analytical chemistry side, mainly based on mass spectrometry, gas chromatography, mass spectrometry, a little bit of liquid chromatography, mass spectrometry. We did that more earlier on. Nowadays we are more focused on applying these methods. We had to go to adventure to the in silico side as well, because these data sets are very big. You had to mine them from the machines. You have to treat them statistically. And to interpret them, you had to rely on genome scale metabolic model. That's the best way to make sense of this soup. So we wrote, not me, myself, but I had students writing programs to help us extract this data in a more efficient way, more high throughput way, analyzes they separate what is artifact from reality, or potential reality at least. And also I collaborate with many groups in a genome scale metabolic modeling of microorganisms. These are many groups in Sweden and Portugal. But what we apply mostly is to microbial system. Find it's a good system to study. They are simple cells. They can grow. You can grow them in a very minimal, very defined medium. So it's easy to do experiments and validate models in these systems. And mainly thinking about industrial microbiology, there's a lot of you can gain from this exercise, because you can use in silico models to do a lot of experiments in silico that you can then select the ones you can go to the lab and do to validate. Mainly for strain improvement, yield improvement, things like that. But the tools that we develop is not only applied to microbial system. We collaborate a lot with the med school in the health science, in biomarker discovery. We can also work with samples in plant samples, insects, and every sort of biological samples, even larvae from mussels in the sea we have analyzed. But mainly not because it's our focus of research. It's people who come to collaborate with us and want to use the methodology we have developed, which is quite applicable to different systems. But most of the research that we focus on is applied to microbial system, mainly in applied side of microbiology. So what I chose to speak here today is one study we have been doing for a long time. It's focused on extracellular metabolite. Why extracellular metabolites? First, it's obvious. You're going to think it's obvious microbial cells, not only microbial cells, most cells secrete metabolites. It's in fermentation in particular, it's known that microbes will secrete ethanol, for example, yeast, some bacteria, lactic acid, some mammalian cells also lactic acid. There are other compounds that can also be they are known to be secreted by microbial system in large quantities, like acetate, formate, citrate, glutamate, lysine, and many others. So industrial microbiology today rely a lot on microbial cells as cell factories to produce these high-value products. Not high value as an antibiotic or anti-tumor drug, but they are bulk chemicals that can be used even to replace some oil-based chemistry. So this is not new. Microbes secrete metabolites. But what is our interest? Our interest is mainly because it's much easier to study metabolites outside the cell. If you want to analyze metabolites inside the cell, it's much trickier. You have to stop the metabolism. These methodologies to stop the metabolism is not simple because you have to work in a very low temperature. You always have a limitation of concentration of cells you can get because it's not like DNA that you can amplify. You have to get a lot of cells to get enough material to detect in an analytical method. So microbiology always point towards looking at extracellular medium to estimate what's happening inside the cell. And that has been done for many decades or century, for more than a century, looking at what the cell produces and try to infer what's the metabolic state of the cell. But with the development of metabolomics, we start discovering that it's not only one or two or three main products the cell produces. The cell actually produces a lot of metabolites. Just before I go into the detail, most of the metabolite secretion has been assumed traditionally that is due to metabolic overflow. For example, yeast cells behave just like cancer cells, that under high sugar concentration they will not respire. They will ferment even in the presence of oxygen. And to recycle the NADH, they overflow the fermentation pathway, accumulating ethanol in case of Saccharomyces cerevisiae, and increase secretion of ethanol. So this has been the assumption that all science and biological science, in terms of microbiology in particular, have assumed that extracellular metabolites is because the cell accumulate that inside, so they had to get rid of it. But when looking at metabolomics, we can start seeing there's a lot, it's very diverse the number of metabolites we can actually find outside the cell. When we stop looking for a list of compounds and actually look at every single peak we detect and consider that a potential compound, we start seeing, for example, in one study I did some time ago, there's a lot of abbreviations here, but these are compounds that were identifying the extracellular media. If you look at the second column here, they are all found in the spent culture media, extracellularly. So this goes a lot of central carbon metabolic intermediates, a lot of primary metabolites. This other study with Saccharomyces cerevisiae in a standard lab medium with ethanol production medium. You can see that many if increased sugar concentration, we have a lot of secretion. All these compounds were secrete, was found in a medium, were quantified in micromole per gram dry weight of cells. So you can see some compounds are very abundant, like pyruvate, succinate, citrate, alpha-ketoglutarate, a lot of amino acids, considering that the media was minimal. There was no carbon, the only carbon source were glucose. So they were leaking a lot of intracellular metabolite. But at that time, everything was interpreted as metabolic overflow. Here an example of E. coli. These are all the metabolites we found in the extracellular medium during a fed batch fermentation of E. coli. And you can see there's a lot of intracellular metabolites of the central carbon metabolism being secreted during the fermentation. Other example, we have another yeast, Candida albicans. You can see here a lot of extracellular metabolites. All the metabolites of an asterisk, they are found in a medium as well, not only inside the cell. So it was clear there was a lot of metabolites outside the cell. Not only these main molecules, the fermentation products that people thought before we started making these big screenings in metabolomics. So the question is how the cell secreted these. And looking for this information, it can be quite surprising that there's very little study about secretion. We know a lot about uptake, of different things, but secretion of primary metabolites is very poorly studied. Basically, the same mechanism that is used for uptake is also used for secretion. In terms of passive, active, the same type of mechanism. But link these mechanisms to specific metabolites is another story. In a review we have made, this is a list of the main, or the mechanisms that are mostly better characterized in microbial system. So you have Corynebacterium glutamicum, E. coli are the best studied bugs in terms of secretion, and they are mainly secretion of amino acids and some organic acids. Saccharomyces cerevisiae very poorly studied the mechanism of secretion. Aspergillus niger. So this is the handful of mechanisms that have been characterized, that has been linked a metabolite to a transporter. We might be missing a few of them, but that's the bulk of it. So comparing to the uptake of metabolites, we basically know nothing about how the cells secrete the different compounds. But based on these mechanisms, they are mainly involved this type of mechanism. But of course, it varies. The same metabolite is secreted by one mechanism in one species and another species by a different mechanism. So there's not much general rule we can assume. A German group also got the attention of these extracellular compounds. I was observing this since 2005. I was very curious, but we have never stopped to study what was going on. This group in Germany, they did quantify, sorry for the resolution here, but they quantified different metabolites in the spent culture medium of different organisms, microorganisms. E. coli, Bacillus, Saccharomyces, and Corynebacterium glutamicum. It's 2 gram positive, gram negative, and yeast. And you can see that some compounds are secreting very large amounts. These are in millimolar concentration. And many of them are, for example, intermediate of glycolysis, some phosphorylate compounds that are considered high energy metabolites, TCA cycle intermediate, a lot of amino acids, intermediates, and final amino acids. So this group went to look at why the cells are secreting this. First, they did a very nice study. They determined if the cells were not These are not artifacts. He said one thing that could be happening is that cells are leaking. Some cells are dying, so they disrupt. So these metabolites we see in the media, they are not secrete actually, they are mostly because cells disrupt. Or during the sample prep, we are losing these metabolites. That's why you're seeing so many metabolites in the spent culture media. So they did the test they test these hypotheses. I'm going to show you the results. And they said, if we don't see any leakage here, so they conclude that we are having all these secretions happening by metabolic overflow through the cell. But then I add that is it only a metabolic overflow, or could we have active efflux? Then me and my students, we went to look at the data we have generated in the past to see if we have any evidence of a different mechanism than metabolic overflow. So that's what I plan to show you the next few minutes. First, let's just clear the air about leakage. These are just copy and paste from their paper. For example, to consider one metabolite they found in considerable amount in the spent culture media of E. coli. So they're assuming the complete release of biomass formed during exponential growth, because they analyzed the samples during exponential growth. So assuming the complete release of the cells of 1 liter bioreactor, they had about 7.8 grams dry weight. If assuming that all cells were leaked, and assume from the literature the maximum concentration that had been found of glyceraldehyde triphosphate in a cell, they conclude the max they could find in the culture media was 4.06 micromoles per liter of glyceraldehyde triphosphate. But what they found actually in the spent culture media revolved the cells much more than that. And the cells certainly were not all of them lysed. To check that, they did an experiment to look at the cell state. They leave the cell growing for several hours until they are completely starved and they start lysing, they start disrupting and dying. It's a very complex figure, but if you look here, you can see that cell dry weight increased, CO2 was produced in a peak here during exponential growth, oxygen being consumed, then it starts stabilizing. The cells start to decrease eventually in the end. But more importantly here, the CN, the cell number, increase and then start decreasing towards the end. And the fraction of damaged cells, they looked through using specific dyes, the cell that only increased towards the end. So actually, it's after 150 hours that the number of damaged cells actually start increasing. And remember, they sampled they harvest their samples in exponential growth much early on, when the damaged cells are much lower. It's here. So it's unlikely that the samples all the cells were leaking, because even in the late stage you don't have all cells leaking still. And even if all cells were leaking, you would expect much less if you only come from the intracellular medium. So it has actually been a production of this compound that has been secreted. What about the technical artifacts? So they sample. What they did to analyze the compounds in extracellular media, that's how we do normally, centrifuge, or we filter or centrifuge, and you look at the spent culture media. So what they did, they take a group of samples that they take some replicates, they take the culture media and extract from the pellet for intracellular analysis. But then they take another group and they wash that group. And then they look at the washing solution and then look at the extract from the cell. But for another group from that group, they wash a second time and then they wash a third time. So they ended up with a lot of 2 washes, 1 filtrate after the cell, and a lot of extracts. What is, making a long story short, the spent culture media is here, reaching a lot of metabolites. after the washing solution doesn't change. So if something was leaking from the cells, we should have seen things appearing after washing. So it is unlikely these metabolites found in spent culture media are artifacts. It might have some artifacts, of course, but the majority of them are genuinely secreted metabolites. And they conclude they were secreted by metabolic overflow. But that's what we question. It's only metabolic overflow. So we went back to some work we did in the past where we have take sample, time series sample, and look at change, measure intra and extracellular metabolites in parallel. There are not many studies like that published that measure intra and extracellular metabolites in that time series event. This one study we did of Enterococcus faecalis in response to oxygen from anaerobic to aerobic. The problem here that we have a little bit of peptone, that is a bit complex, but most of carbon comes from glucose. And you can see that a lot of metabolites have an apparent metabolic overflow. Like you see intracellular from anaerobic to aerobic, you have an increase. You see an increase in the media, a decrease, a decrease. It's like mirroring what's happening inside the cell. What has been always assumed, most of the metabolomics studies. But we have a few exceptions as well. For example, phenylalanine here, you have a drastic decrease inside the cell in response to oxygen, and you see that seems the cells are just secreting it to the extracellular medium, popping out. So in this case, we can't say that phenylalanine, for example, are being secreted because there's accumulation inside the cell. So the theory of metabolic overflow gets a bit complicated here. The same with valine, the same with levulinic acid. It could be because there's a little bit of peptone in the media, we could say that maybe there was a less uptake under aerobic condition, because the cells didn't need under aerobic condition that those amino acids they can synthesize. So they reduce inside the cell and they didn't uptake more.
Sorry, what does peptone do?
Sorry?
That's a bit of a coincidence. Silas, could you give us the definition of metabolic overflow?
I can go back very quickly. The easiest way to understand a metabolic overflow is this classical fermentation situation, the crabtree effect. The cell has to overflow the fermentation pathway to recycle NADH. So what means that the cells are increasing the flux through this pathway, and this pathway ends up in a product that accumulates in the cell. So that's why these are secreted. That's what is believed to be secreted. So a metabolic overflow is assuming that something is secreted because it's been an accumulation inside the cell, an overflow from a pathway that produced that compound. Can you follow?
So there's no mechanism, it would just be, otherwise it would continue to increase without limit. Is that the idea? Inside the cell?
Yes.
Overflow.
Yes. So that's why there is a secretion. So the secretion is justified based on an overflow happening inside the cell.
So there's no feedback mechanism, negative feedback mechanism, and you have to shut off the...
For some compounds are, but some of them, like in this case, that are crucial for the cell to recycle the redox potential by the recycling ADH to keep glycolysis flowing, and because it's the only way the cell can produce ATP here. It will be not good for the cell to regulate that, otherwise they will stop growing. So one mechanism that has been favorable is to keep that overflowing and shipping it out of the cell.
So the cell absolutely has to get rid of it?
Yes, because... You have a question?
So
Let's go back to that slide. So
peptone may have confounded some of those data, so it wasn't clear what the role of peptone.
if you think that peptone is a protein hydrolysate, it has a lot of mostly amino acid peptides, but also some other metabolites. So if you think that these amino acids could also be found in the medium. You remember these are two situations that this is aerobic, this is anaerobic. What it could have happened, these are metabolic overflow. The ones that are against metabolic overflow theory could, in this case, could be justified that the cell just didn't require much phenylalanine under aerobic condition, what would be possible, because aerobically they can synthesize, they have more energy, they can synthesize all these amino acids anyway. So they could have less uptake the phenylalanine from the media, so we see an increase, a higher concentration in the aerobic condition in a spent culture media. So because it is not clear if they are secreted or not, because they are already in the medium. So to avoid this confusion, we compare another study that was no complexity in the medium. This is E. coli during heterologous protein production in the fed batch fermentation. Glucose, the main carbon source. There is valine, so the only thing is valine. We cannot make any conclusion about valine because it's providing the media for this. And the cells are supplied with this inducer to induce heterologous protein production. Here, again, we have a few conditions we have seen throughout different hours here. You can see some clear trends that are what is expected from a clear metabolic overflow inside the cell. Increase here, increase outside the cell as well. When you have a decrease, there's not much example of decrease in this case, but you could also have a decrease inside the cell that will decrease from the outside. But again, we have some condition that goes against metabolic overflow. Metabolite data can be quite variable, as you can see here for the replicate. So what I ask you to pay attention is not always in the same end, beginning the end. Sometimes you have to think in these 2 points here between 5 and 11 hours, you have a clear increase in the ecanoic acid. And if you look here, you can a clear decrease in this point, that point is statistically significant. So if the figure here is because some of the points are statistically significant, you see a different trend, a decrease inside the cell, slight decrease, but a considerable increase outside the cell. The same goes with benzoic acid, clear decrease inside the cell, a shift towards increase here, and the last sample there was a decline. But if you look in these 2 points, it's a clear thing. Without going into more details here, let's go to another case. Candida albicans in response to farnesol. Here's minimum media, there's no other organic compound, just farnesol, there is a quorum sensing molecule, a high alcohol, and glucose. And here, there are some clear, there are 3 sample points, 3 hours, 6 hours, 12 hours. You can see here typical mirror situation. What happens inside the cell seems to be reproducing outside the cell. In general terms, if you think. But again, we come to some clear situation where there are opposite effects. For example, isopropyl malic, I think, is one of the most beautiful data we have in terms of inside the cell, clear decrease, outside the cell, clear increase. So you can't say there's accumulate inside the cell, but even though the cell still get reading of this compound. This is a key intermediate for the branch chain amino acids. So you would expect to be important metabolite to be kept inside the cell. Citramalic, there's an increase, then decrease. If you look at this point in these last 2 hours, you have a clear sharp decrease. If you look at this 2 hours, last 2 hours, a sharp increase. Actually, all the 3 hours are a sharp increase. The same is here. So, based on these studies that were not designed to test these theories, we came up with this hypothesis that metabolic overflow may not be the only cause of intracellular metabolite secretion during growth. So we decided to do a target experiment that we published last year to see if we can predict intracellular metabolic state based on extracellular metabolite. Why this title is very provocative title for the field? Because there was a lot of reviews and opinions defending the idea of using extracellular metabolite profile to predict intracellular metabolic state, or even further, distribution, a flux distribution inside the cell. So that's why you want to say, well, can we really do that? Do we really know how the relationship between intracellular metabolites to make this prediction? So we use a very classic example, using Saccharomyces cerevisiae, a lab strain that is well studied worldwide, and a typical metabolic perturbation from anaerobic to aerobic, where there's a classic change in metabolism. Time series experiment, we use continuous culture to keep the growth rate constant in both conditions. And we use a low concentration of glucose, so there's no carbon catabolite repression, no crabtree effect. This was the experiment was designed. Start anaerobic, a batch phase. When you run out of sugar, we start feeding at 5 grams per liter of glucose. Minimal media, there was no other carbon source, only glucose. Keep 2 residential time in the bioreactor to make sure we have a steady state. We took 3 samples for intracellular metabolite. And then we perturb the system by adding oxygen, air, allowing it to become completely aerobic. And then we took samples every 20 minutes to see the response of the cell to the oxygen getting inside the cell. And of course, we disturb the steady state. Because we disturb, we don't have replicates here. But then we reach another steady state. Remember, the growth rate is still the same in both conditions. We have higher biomass here because aerobic you have more biomass production. And we wait 3 residential time to guarantee we have a steady state, and we took samples again. So just to validate the experiment were done, run as we expected, we have a clear difference in metabolic pathway activity comparing anaerobic with aerobic. You could see here an increase in a lot of typical pathways that are aerobic metabolism, TCA cycle, increase activity, pyruvate metabolism, glycolysis, flux increase, all these. With a few exceptions that you have a decrease like biotin metabolism. That is more crucial under anaerobic condition. Some significant metabolites to assess key pathways like citromalic associated to the branched debasic acid metabolism. Pyruvate metabolism, lactic acid decrease the concentration in the fermentation reduce in response to oxygen, TCA cycle increase, and so on. Classical response to Saccharomyces cerevisiae, we compare with some transcriptome data using the same setup of experiment, the same strain. It was very comparable. So then we look at the metabolites. Well, I'll show you a few cases, a few scenarios. The first scenario is only intracellularly there was a significant change. In this case, it's the red line. So in extracellular, we either did identify the compound or they were not changed significantly. So you can see here examples that are lysine, for example, they were only found intracellularly, has an increase over time. In response for the 2 steady states, a clear increase, but we never detect it outside the cell. There are cases like hexanoic acid, you detect outside the cell, but it's not significant, it's very variable, the data. Inside the cell, it is significant, there is a decrease between the 2 steady state. Remember, the transitional state is not steady state, so it can be very variable. The most important are the 2 steady state points that we have replicates as well. So that's the case that we expect, of course, we don't expect to find every intracellular metabolite also outside the cell, so that's okay. Another scenario where you find metabolites that are both inside and outside the cell, and they change significantly both inside and outside the cell. And we find, on the other extreme, metabolites that are only significant change outside the cell. And that's worrying if we think about using extracellular metabolism and metabolites to predict intracellular metabolism. Because if only outside change and not inside, how can we say we can say the outside profile is reflecting what's happening inside the cell. A clear example you can see here, there's a significant decrease in GABA concentration outside the cell, but inside the cell the changes are not significant. Actually, if you compare the two steady states, they are identical basically. So if we just look in outside media, we'd say, well, there was an increase in GABA metabolism inside the cell, or in production of GABA, but actually there was not. Well, there are different ways to approach that anyway. Glutaric acid, a clear increase outside the cell, no significant change. And more puzzling, you have a situation where you find metabolite only outside the cell. We don't detect inside the cell. How can we explain that? That's a bit tricky. All the threshold level inside the cell, toxicity, maybe, perhaps, is too high that it has to be produced and secreted immediately. So we can never detect enough of it. But it's very puzzling when you find compound outside the cell, you don't find inside the cell. So in terms of patterns, we found those typical mirror pattern, metabolites that decrease outside, decrease inside. Most are decreasing here. There's no case of increasing in this situation. Or there is a decrease in response to oxygen or that metabolite, which is expected, because if you have oxygen, there's more incorporation of these metabolites into biomass, so you expect a decrease on these intracellular compounds. And what is interesting, that also reflects outside the medium. But then you have situation like you have opposite direction here. I think one clear here, aspartic acid, you have a decrease inside the cell, and here you have a increase, a significant increase between the two steady states. And remember, this is a minimal media, there was no amino acid outside before. So this was secreted for the cell, literally. The same for the canoic acid. Benzoic acid is a significant increase despite the variation during the transition state. But inside the cell, there are decrease. So that's an indication these compounds are being pumping out of the cell. As I told you, there are some situations that intracellular change, but extracellular did not change. For example, intracellular, there was no change here, no change there, no significant change, but extracellular there was. So we cannot blindly assume that everything that happens outside the cell is also happening inside the cell, as we can find here in this another case here that is in the opposite direction. How to interpret that? Well, we're still thinking about it. Some authors, for example, that consider everything is a metabolic overflow, they think that cells use the extracellular media as a storage space. Giardina found some enzymes in the extracellular media using proteomics. They also suggest that the extracellular space could be used as a storage space. And they said the metabolic reaction could take place in extracellular media. So some of these metabolites that are only found in the extracellular media perhaps could be forming there. That's why we don't find inside the cell. But I think that makes sense for a pure culture. How that works in a mixed population, I would wonder, in terms of the advantage for the cell. This group also suggests that the enzymes these are metabolic enzymes found outside the cell, may have a different function outside the cell. Doesn't make the same type of reaction. So maybe the metabolites also have a different function. Cell regulation. We know there are metabolites working cell-cell communication, codon sensing. So how much of these primary metabolites I also use as codon sensing and communication? That's a question. We don't know. This group said the metabolites become toxic under specific condition. Yes, this is an old concept. It seems to be clear for some compounds like benzoic acid. It seems to happen in all systems we observe. Benzoic acid seems always to be secreted. It never accumulates inside the cell. Some fatty acids also, and they are known to be toxic. So there are a good degree of truth around that. But it's only that is driving these cases that are against metabolic overflow. That's a big question. Just to sum up, these are the main patterns we found. Mirror pattern, intracellular, extracellular, identical, going up or going down. When it's going up, it's a clear case of secretion, metabolic overflow. Here, a clear case of uptake, perhaps, if this compound is also present in the medium. Here you can see one of the best describing examples in Saccharomyces and E. coli there. Other type of pattern is when there are opposite directions, intracellular increase, extracellular decrease. I would say that's a clear case of uptake. These are intracellular decrease, extracellular increase. I would say that's a clear case of active efflux of that compound without metabolic overflow. And there are other patterns that is not so easy to explain, like things change intracellularly, but there's no change extracellularly. So if you use the data from extracellular, you will not observe that change. In other case, things don't change intracellularly, only change extracellularly significantly. So you think there are things change related to that compound inside the cell. It'll be difficult how to relate this information to that. But what can be even more puzzling is when we don't find inside the cell and you find things outside the cell only. And as I told you, there are groups working with modeling and trying to use extracellular, or defending the idea of using extracellular profile metabolites to predict intracellular metabolic state or flux state of the cells. And that's what we find. Are we ready to do that? I had a student that's worked with genome scale metabolic modeling, and he did that comparison in 4 well-developed genome scale metabolic models, ecometric models. And you can see that the models can only secrete under the same experimental conditions that these were done, published, a very small number of metabolites. While, for example, in this case, Saccharomyces, when the Saccharomyces you can detect all these metabolites experimentally, the model only predict this small amount using flux balance analysis. Using flux variability analysis, you can actually have a little bit more secretion, but there are compounds that are not allowed to be secreted or impossible to be secreted by the model. And there are compounds that are even more worrying that are not found in the model because they are not linked to an enzyme reaction yet. They are metabolites that we routinely find them, but because there are still genes that we don't know the function, so we don't know which reactions are producing or using those compounds. And you can see that applies to different organisms. E. coli and Saccharomyces are the best models, and still we can see that we can predict very little. What's the impact on the fitness of the model? Not much. If you think in terms of carbon, these metabolites we count by less than 1% of the carbon, if you think. I think the most important metabolites are here. But still, if you are using these models to optimize and strain, to optimize strategy to increase yield, you may fail to predict accumulation of a compound that can be toxic to the cell and affect the cell. So you may reduce the ability to your chance of success by optimizing the strain. In the same, if we want to use extracellular metabolite profile data, that's much easier to obtain, much quicker. To be able to use that to predict intracellular metabolite data, we still need to understand much more about how these cells are secreting these compounds, why they are secreting these compounds, etc. So some very key conclusions we think we can draw from these studies. Microbial cells secrete a lot of compounds that it was not very clear 10 years ago. I think from these last 10 years, we have been realizing there's a lot of compounds leaking from the cell for different reasons, metabolic overflow or not. So many, maybe most, we don't know, in our experiences, about 50–60% secreted metabolites seems to be secreted due to intracellular metabolic overflow, following that pattern of mirroring what's happening inside the cell. But there's a great proportion that doesn't follow that. So we think that, we defend the idea that we still have to study more to understand these if we want to improve our ability to model cells, metabolism, and also to use extracellular metabolites for predicting, as a tool to predict metabolic states. I think the presence of metabolite in metabolome indicates that relevant reactions should be included in network reconstruction, what is not at the moment. So, well. So I just had to, this work was collaboration with a lot of groups, different people. As I said, I have collaborated overseas in the metabolic modeling part. My group, we have previous former PhD students, currently postdocs at the university, and the current PhD students. The funding, of course, we never had the funding direct to do this. We leverage these studies through other funding that we get in the lab. It's still hard to get funding for these basic metabolic studies. And of course, my group, this is an old picture. They have changed a lot. And I thank you for attention. If you have any question, any suggestion, I'd be happy to answer.
Thank you, Silas.
Okay.
Questions?
Yeah, I just have a question. In at least one case, you showed where the metabolite was decreasing extracellularly but increasing intracellularly, and you said that's probably a case of uptake. But
It could.
Yeah, but if that wasn't an experiment where you had minimal media, where it was just valine.
Yes, but if you remember, that was a continuous culture. There was a situation, and then we had a perturbation, and then you have another state.
I'm thinking of not your design experiment that you designed specifically for this.
No, the other one, but all of them are time series. Yeah. So you have a situation before and after. Yeah. So if you have that compound out, and the next time you have less, it could have been uptaken, isn't it?
So you're thinking like maybe, I'm just, like if it didn't come from your original media components, like maybe you're saying like it was produced before your first time point, but then...
Yes,
okay.
Yeah, that's what I'm saying. During the first time that we sampled, that was in a high level. The second time we sampled, it had decreased. So it could be because the cells uptook them.
Like they produced them and then took it back. Okay.
Mm-hmm.
I didn't hear you mentioning, when I think of yeast, I think of ecosystem engineering, and I thought maybe that's one possibility that would be really cool to explore. Maybe they're sensing there's some cue in the environment, they're making them secrete some other metabolite that's going to further change the environment, that's going to be beneficial for something?
There's a lot of possibilities.
Yes.
All these experiments are pure culture. Very artificial, of course, in terms of if you want to see what's happening in nature. But definitely, that's the question mark. Is it a higher degree of communication in cells involves primary metabolism? Yes, co-culture or population culture could comparison.
Those that show weird patterns.
Exactly.
Those are the ones that, that's very interesting.
That's I think the aim was to show you that we know very little still, and showing there's a lot to be answered. And I think the main contribution we have made is that, look, there are a lot of things come out and back the cell, and they don't follow what we assumed as follow, the metabolic overflow theory. So I think that.
This is a rather naive question, but I'm wondering what do you mean by steady state?
Steady state means the cells are in a continuous growth. They are not limited by substrate. So they are growing the highest speed or the highest growth rate at that condition. So that's equivalent to the exponential growth.
So it's an exponential.
It's a continuous exponential growth. But because we keep feeding and taking out media, we keep the concentration constant. but the cells are multiplying in the
So the
highest
medium is
rate.
constantly being changed? Is that what you're saying?
There are constantly new media being fed to the bioreactor. In that was only the last experiment with Saccharomyces that we
The cell
did.
number is not steady, but some other things are steady.
The growth rate is steady. Is the growth rate, the rate of division.
The cell number is going up exponentially.
Yes. They don't build up in the reactor because we take them
But
out.
there's no accumulation in the medium because it's constantly
Yes,
being
no,
changed.
no. Okay, that's the best way to study primary metabolism. And else we don't have build-up from the media. So, because if you build up things from the media, you change the conditions, you change a lot. So in this case, you maintain constant the situation. Not the transition state though. So that's why the transition stage, you see that some of them are very, very buoyant.
Yes.
There are, I guess, mammalian cells that will either secrete or take up a substance in order to maintain volume or, in the face of an osmotic challenge. If they're shrunk, they will take something up to draw water in again. Some of that looks like could be explained by that.
Yeah, that's an interesting point, actually. We don't think much about that in microbes because of their cell wall. But indeed, it could also apply to them, definitely. Maybe I'm uninformed about this effect, but it's known that osmotic shock produces leakage of compounds in microbes. So yes, it could be an interesting process to consider, to look at the effect and see what could be happening.
So would genetic modification be a useful way to study some of these things? So because some of the perturbations you're doing, so you're switching from, let's say, an anaerobic culture to an aerobic one, and that's hitting a lot of your metabolic reactions. Would it be possible to to target, let's say, one particular enzyme in the pathway?
You could, I think, you can provoke specific accumulations to see how the cells behave in terms of would they keep the metabolite inside or they would. And you can study some threshold of secretion and can be used also to study secretion mechanism, which is another area that you work with genetic modification to knock out genes responsible for potential transporters to see if they are the ones linked to that. You see, there's a lot of scenarios to study, and genetic knockout and even overexpression can be used to provoke this situation. I think modeling, if we could incorporate more transports that is known, we could try to do more in silico experiments about that and seeing what could affect the secretion, then try to validate. Because analyzing extracellular media is much easier, it's very quick, cheap, than look intracellular. So if you can do a lot of simulations, and then we can go and test that in the lab in a small scale and well played.
But I guess, I mean, with what you mentioned, one of the problems with building up models,
Yes.
in silico models, is you don't know what the transporters are.
Yes.
So how do you...
Yes, so there's another area that has to be developed in parallel, that is the study of transporters, which is, I don't think, is so linked to the metabolomics, but would be of great help together to understand what link transporters with specific compounds.
What concentration do you need to be able to use your assay methods?
Depends. So we need, depend, for extracellular, we work with very low concentrations, like
What?
we're working about nanomoles level. We use culture media directly, like 100 microliter of culture media is enough. So we can do multi-well studies. That's why the beauty of using extracellular. You can do very micro cultures and makes life much easier to do a lot of testing. If you want to go intracellular, then you have to do big
Yeah, I'm thinking
cultures.
intracellular, yeah.
Intracellular, you have to get bigger. 20 milligrams dry weight of cells, which can be quite challenging for some species at least.
I thought Google was the first to store information in the cloud, but I see that these cells do too. They dump information out and it's in the medium.
It's a good analogy. It could. I think there's more information in the metabolites that maybe we think, we understand. Yeah.
Any other questions? Right, let's thank Silas for his
Thank
talk.
you.
There are refreshments.